Last week we looked at the teleologic argument from the perspective of the universe. And what we did was we concluded that purely scientifically, I get nothing from a biblical account, but purely scientifically, that the universe had an origin, that not only did the universe have an origin and apparently spring out of somethingness from utter nothingness, that suggests and implies that there was something outside of nature that brought it into existence. That's the cosmological argument. But also that when you look at the universe, it seems very precisely tuned and exact. And Joe, can you get our TV going here? It seems very precisely tuned and is such a way with constants in things like the gravitational forces, the distance of stars, the motion of planets, the fact that the moon is around the earth with just its type of forces, the tilt of the earth, and just all kinds of things. There's over 100 of these little constants that, without them being all precisely, exactly what they are, it would be incompatible with life on earth. Now, we're going to use that same understanding and argument to take a look now at first life. And if you'll give me just a moment to get the technology up and running, we will get started with that.
So I can see around you.
Sorry. When we look at life, we have to understand that the current thinking about how life came into existence from evolutionary methods, it requires that that the person adopting the naturalist or humanist view, that life was generated spontaneously from non-living chemicals. I'm sorry. It's not going to let me do this. It won't let me play my PowerPoint and then open up AirPlay. I have to open up AirPlay first and then start the PowerPoint. Okay.
It's amazing it ever works, if you think about what you're
How
doing.
come it's not working, Joe?
like when you were 15, did you ever think
I
that
clicked
you could like have a
AirPlay,
computer
library.
this big that you carried in your pocket, and then it would broadcast signals to your TV, so you could actually watch your phone
I
on your
was really
TV?
thinking like Back to the Future, and they're like, oh, look at this stuff, and then all these things, you know.
So,
It's amazing.
so what it requires is that life was generated spontaneously from non-living chemicals by completely natural laws, without any intelligent intervention. Now, this, quite frankly, in the days of Darwin, seemed quite plausible, because although they would acknowledge that the cell was very complex, they did not know in the mid-1800s what we know today, simply what technology has shown us is going on inside of the cell. So before we ask the question about that possibility, what I wanted to do today was to take you on a little journey inside the cell. Now, one of the great blessings about this is that there will be no test. at the end of the class.
Yay!
And not only that, but I'm going to be showing you a lot of stuff, and I'm going to be making analogies to things that we know and understand well, but there's a lot of big words, and you don't have to remember them. The only thing that I'm trying to do is to impress you with scratching only the surface of what's going on inside of each little cell, and to know that there's 100 trillion of these suckers inside of your body doing all of this. Okay? So we are continuing on the teleologic argument. Now, in the last 60 years, we have, well, I just want to remind you here, what is science is a search for causes, and there's two types of causes. There's non-intelligent causes, natural causes, and there are intelligent causes. We might have to deal with a little bit of that because there's a delay between when it pops up there and what I see on my little screen. So I'm just explaining that to you. And what we've done is we've looked at what the causes of the universe. What is the evidence for the causes of the universe? That was 2 weeks ago. And today we're going to look at last week, and today we're going to look at the causes of life and what the scientific evidences are of that. Now, in the last 60 years, biologists have been able to open up what is inside the cell because of advances in microscopes and the technology of how we can separate cellular components in the laboratory. And as we peek inside the tiny world of the living cell, we discover amazing complexity and awesome design. If we consider even the very simplest life forms, like the amoeba, I want to take a look at some of the structures that are in here. Now, all of this works together to keep this little thing alive and to keep it replicating. You've got the nucleus, which is filled with eukaryotic DNA that has the coding for all of the processes that go on in the cell, the coding for every protein, every lipid, everything that's going on in here, every process that allows that cell to live is coded for inside the nucleus. It's got a membrane. The membrane is an amazing structure because if you watch an amoeba, it's never fluid. It doesn't stay the same. It's constantly changing with these little things that we call pseudopods, fake foots, okay? Fake feet that kind of ooze around as this thing goes to find little chemical food or little bacteria that it can digest and eat, and it will wrap around them. It's got a food vacuole. If there's a little bacteria right here and it kind of wrapped around it, it would bring it inside into this little food vacuole. That membrane breaks off and becomes a different structure inside the cell, and it's got to know what it's doing and how to do that. There's other little vacuoles and little membrane enclosed structures that will mix with that thing and start digesting it. There is ectoplasm and endoplasm, endoplasm being the jelly-like substance of the cell. There is processes going on that allow for its replication and its movement and its search for food. It can go and recognize current of electricity, and it will go towards one side of the current as opposed to the other side. It can recognize light, and it can go away from bright lights, but yet it will go away from darkness to go to a medium type of light. And all of these things are happening inside the cell on a chemical and molecular basis. Now, I want to review some cell anatomy and go over some details. Okay? Inside the cell, you have this thing called the nucleus. And inside the nucleus is the genetic material, DNA. Now, I have this picture here because I wanted to show you this structure of DNA. And I was gonna talk about this later, but I just want to point this out. If you took one of your cells, okay, and these are, atoms making up molecules that are super, super long here. This is a DNA molecule. It is a double helix. It's that kind of twisted ladder appearance. That's what a double helix is. And it's really, really long, okay? We have 26 chromosomes, and the way that chromosome is put in there, have you ever taken clothes and just kind of mashed them into your suitcase without folding them up? Okay. Now, if you do that, you find that if you take them out and you fold them up really nice and compact, you can fit a whole lot more clothes than if you just stuff them in. So if you take all that long DNA and you just jam it into the cell, and I'm going to tell you this, okay? If you were to take the DNA in your cells, just one cell, and you were to pull it and stretch it out to as long as it could go, okay? Every DNA inside, it would stretch out to about 2 meters, okay? About 6 feet of
cell.
DNA of this molecular thing.
Every cell.
Now,
Yeah, but I mean that's just one
Times-
cell stretching out.
and now, if
Yeah,
you took all the cells of your body, and you stretched out all its DNA end to end, you could go from Earth to Pluto and back 17 times.
Oh,
One
Wow.
person. Okay. Now, the reason that's amazing is you're stuffing all of that into a nucleus, which is measured in microns. Okay. I mean, it's tiny. Okay.
That means God's a good packer, right?
He's a good packer. That's exactly
great
what I'm
one to go on
going at.
vacation with.
That's exactly what I'm going at. So not only is all this information in there, but there's lots and lots and lots of it, and it's stuffed into a tiny area. And think about what it takes to get your clothes packed in a suitcase. That takes all by itself an intelligent way of saying, how can I manage this a little bit better to get all this stuff to fit in here? And what you do is, when you add these little things, there's these proteins called histones, and when they add to the DNA, they cause it to curl up into this configuration that makes it much tighter than if the DNA was in there all by itself. And not only that, but those things twist around and they curl up into this little X-shaped structure that we call a chromosome, and all of that happening 26 times in one single nucleus. All right. So it is just an amazing thing all by itself. But this is a basic eukaryotic cell. And I want to just run through this real quick. Cell Anatomy 101. So,
What's eukaryotic? Like normal,
so
like yours, right? Like
yes, we
that's our
have
cell.
eukaryotic cells, and there are prokaryotic cells. And so bacteria is not a eukaryotic cell. Bacteria is a prokaryotic cell. Plants and animal cells are eukaryotic. And I guess a very basic understanding of it is the higher, more complex forms of life have eukaryotic cells. So there is a plasma membrane, which is this amazing lipid-rich sac that knows just what to let out and what to let in. Very amazingly complexly designed. It is not just a wall of fat that holds in a sack of mush. It has got ion channels. It conducts electricity. It can recognize chemicals around it that makes it do different things to either pull it in or push it out. Okay, amazing structure. There's cytoplasm. Cytoplasm is kind of the jelly-like stuff that everything is living in. There is the nucleus, which contains all the information and the blueprint. It's the brains of the cellular operation. And inside the nucleus is the nucleolus, which is where all of the information is densely packed. There is this chromatin. Chromatin, if you remember our picture with the DNA kind of unwinding from the chromosome, that would be what chromatin is. Kind of slightly unwound DNA you think about all the little stringiness of the double helix kind of coming unwound from it. That's what your chromatin is. Chromatin is the stuff that's available then to transmit the message that it carries into other things that the cell is doing. Then you've got the endoplasmic reticulum. So you've got the rough endoplasmic, this yellow stuff with little grainy dots in it. That is rough endoplasmic reticulum. That is the protein making conveyor belt and machinery. It is the production plant for everything else that goes on in that cell. There is also smooth endoplasmic reticulum, which makes the lipids and the stuff that will end up being in the membrane and the membrane proteins. The membrane contains a lot of proteins on it that allows it to do its functions. And then there's something called the Golgi apparatus right here. The Golgi apparatus is the storage plant. So what it does is it's responsible for packaging, storing, and then transporting the proteins that are being made to different areas of the cell, and it basically manages the manufactured products from the rough endoplasmic reticulum. Then you've got the mitochondria. The mitochondria is the power plant of the cell. The mitochondria is where you actually get the chemical energy made that is actually used to power that cell so that it can have a production plant, make proteins, move everything around, create different shapes, move things in and out of the cell. It needs energy to do that. The energy is produced right here by taking chemical energy and turning it into a usable chemical energy called ATP inside of the cell. Evan's going to learn a lot about that in biochemistry very, very soon.
What's ATP stand for?
Adenosine triphosphate.
I'm glad I asked that.
You don't know that.
Yeah.
It's part of the Krebs cycle, which if you've ever taken a biochemistry class, it's like, oh wow. Organic chemistry.
Okay, so the Krebs cycle is another example of a beautifully designed chemical pathway that reliably produces energy that these living forms can then use to do all of their cellular process. It's just a beautiful, beautiful process. So mitochondria, lysosomes. Lysosomes, these little blue things is represented in this picture here, are kind of the stomach of the cell. They break down proteins and lipids and carbohydrates, and in white blood cells, that the lysosomes are what's fighting infection for you. So if you've got, for example, a pneumonia, white blood cells go into that area, they find the bacteria, and they eat it. They chomp it up there. And then when it goes inside the cell, it's in a lysosome where it gets digested. When you have a big bunch of these white cells eating up that bacteria, when you see it, that's what pus is. If you ever had a sore that had a pussy drainage, that's the white cells eating the bacteria. Okay, so that's the lysosome, the stomach of the cell. Then they've got this thing called centrioles. The centriole is an interesting structure that is important in replication and cell division. It separates chromosomes and is an important aspect of that cell reproducing itself. And then you've got these things called microtubules. The microtubules are a part of the movement of this. They do a lot of different things. They help the cell in movement. They are important in nucleic acid and cell division. Nucleic acid is the components of DNA. And then it also helps in cell organization and intracellular transport. So kind of movement inside the cell, movement of the cell itself, and things moving along these microtubules, as well as replication. They have lots of different functions inside the cell. So anyway, you have got a really dynamic and complex set of activities going on here. Now, I want to take a look at some of those dynamic activities and look at what happens when a cell does one simple, and I put simple in quotes things, and that is, how does it make a protein? How does a cell itself make a protein? Because these proteins are, everything to the substance and functioning of the cell. it is the major part of how it does all of its operations. It makes proteins as the conveyor belt of the manufacturing process. The proteins are on the surface of the cell. The proteins are the substance of what is made inside the cell. Proteins are things like those histones that allow the DNA to wrap itself up into a tiny configuration. Proteins are not everything, but proteins are the major portion of what a cell makes so that it can perpetuate its life. So the first thing that happens in making a protein is something called transcription. You remember you had your DNA, and it has to know that a particular protein is needed. So when it realizes that, however it does that, it unwinds itself from the chromosome so that the gene that codes for that protein can be exposed. Next thing is that DNA, as you remember, it's like a double-stranded ladder. It has to unzip. It has to open itself up because the message is on the inside. So the first thing that happens is there is a zipper, okay? The zipper is called helicase, and that helicase attaches and allows the DNA to unwind. The next thing that happens is you have a protein. So protein helicase, protein RNA polymerase attaches. RNA polymerase, poly means many. Polymerase refers to the things that allows you to make something. Yeah?
No.
Oh, okay. So, so it attaches to the DNA strand, and it starts to make an RNA strand from the DNA strand that is coded in the DNA. So I'm going to talk more about how it does this, but just what you need to know is that the code is on the DNA. You see in this blue here. This RNA polymerase comes in here, and it starts making RNA from the template of the DNA, and you get this RNA strand. Now, this RNA strand, we would call that a messenger RNA, mRNA. Why? Because that is the message that is going to make the protein. The code, the information is in the DNA. The RNA is the thing that is going to go outside of the nucleus and actually start making the protein. Okay. So now, this is the next thing that happens. After it gets all the way through its sequence, after it gets all the way through the sequence of DNA, so that we have a complete strand of RNA, what happens next is it separates, okay? And then your DNA can wind back up, okay? And then the RNA gets a whole bunch of modifications preparing it. For example, you can get polyadenylated, you can get capped, you can get spliced. It gets processed. There's an actual processing that occurs of that message to make it just right to make the protein. So after that happens, you then have the process of translation. Translation means you translate this message, the message RNA, into an actual protein. That's what translation means. So the RNA, after it gets processed, it leaves the nucleus through the membrane of the nucleus, and it goes into the cytoplasm. That thing then finds its way to the rough endoplasmic reticulum, which is the processing plant.
So it's kind of like the DNA makes the mold, and then the mold goes to the factory.
That's correct. A copy of the mold.
Right.
Okay? The mold doesn't change. A copy of the mold then goes to the factory to then get filled up, okay, after your mold is done, to make your product. Okay? That's a good analogy.
Actual biological factory.
It is an actual biological factory. So now we're on the rough endoplasmic reticulum, and that messenger RNA hooks up to a ribosome. The ribosome are the little bumpy stuff on that rough endoplasmic reticulum that makes it be called rough. Okay? So this here represents a ribosome, and this strand up in here is our message RNA that has been transcribed from the DNA. And then you've got these little transport vehicles, And we call these transport vehicles, they are also RNA. They're tRNA. We call it transfer RNA. Why? Because they're transferring the building blocks of the protein to the messenger RNA to put it all together. So think of it, the ribosome, like a conveyor belt, okay? And along that conveyor belt goes the molds. And the transfer RNA has very specific little 3 peptides, okay? 3, nucleosides they're called. 3 nucleosides that match perfectly with the message. So only one transfer RNA can fit with a particular message on this message RNA. So on the ribosome, you've got 3 different spots. Okay, you have got an A spot, a P spot, and an E spot. You can see this here: an A, a P, and an E. Now, what it actually stands for is an amino acyl tRNA binding site. Here's the A, amino acyl. You've got a P, which is the peptidyl tRNA binding site, and E, exit. I'm going to give you a better way to remember that. A is the accepting site, P is the processing site, E is the exit site, drop off. Okay? So here we go.
That's amazing.
Little, this here transfer RNA has got its little amino acid. Proteins are made of amino acids. You hook up a bunch of different amino acids together, that gives you your protein. Okay? So it comes to the A site. It attaches the accepting site. It finds by the perfect binding of these here. This codes for only one type of tRNA. The one that this AAA codes for a UUU. They always match up just like that. And then this particular transfer RNA with the UUU only carries lysine. Never carries anything else. It only carries lysine. So if there's an AAA on your messenger RNA, you know that you're only going to get a lysine there. So boom, that thing boom, hops up, goes along the conveyor belt to the next thing. This one pops off. You've now got your lysine moving over to the processing site while the next one comes in. Now, while it's on the P site, I call it the processing site because that's where the amino acids hook up. So now, for example, the glue and the val, they hook up here. They move off. The little thing pops off. And the next one comes in where it can get processed and attached. You follow? That is how proteins are built. And it goes along this little thing here. It goes along this little thing, just hooking them up one at a time on the conveyor belt, processing it along the ribosome until you get all done. And there's these are called, by the way, these are called codons. Okay, that's a codon. 3 units because the transfer RNA has 3 units, and they've got to match perfectly. And so this is the anticodon. A codon and an anticodon connect perfectly. And there's a codon at the end, which is a stop codon. So the ribosome says, oop, I read the stop codon, everything separates, and you've got your protein completed. Now, every single protein, and there's thousands, okay, of these proteins that have all different functions. Those proteins, because of the type of molecules that they have little positive and negative areas on there, and it causes them to fold up in a very specific position. And that folding up into its very specific position is what allows it to do its particular job. The ribosome is made up of several proteins. They have to fit in just exactly the right way to suddenly now be able to do this specific process. It has to be specifically designed in a specific configuration to be able to do its job of actually making proteins. Okay, that's just one example of thousands. All right.
So the ribosome itself is made out of what it's creating.
That's exactly right. The ribosome, this could be making a new ribosome. This could be making something that helps digest food. This could be making whatever. I mean, there's just thousands of these. It's just, you just can't believe it. By the way, I I was a biology major. I majored in cell biology. This was my passion when I was 30 years ago.
Well, if you don't mind me asking, do you mind if I put you on the spot for a second?
No,
Now, were you an atheist back then?
absolutely.
Okay, how did you mesh that? Because would not the odds of that being possible with the precision of that process to have taken place from just some random explosion, would that not be astronomical in terms of odds, almost to infinity?
Yeah. So I didn't think of it in those terms, but we're going to address that.
Okay.
Yeah, that's the mighty conclusion at the end of class.
Oh, sorry.
All right. So what I want to do is I don't want you necessarily to remember all of the terms and what's going on in translation and all this. I want you to understand that what's going on inside the cell is amazingly complex. It is specified complexity. It has very specific purposes with machinery and processes, highly designed processes, as well as just amazing complexity. This is just one little thing. The complexity of the mind is mind-blowing, I think, to the same level that the complexity of the universe is mind-blowing. This is the universe complexity on a molecular level, whereas the cosmos are on a cosmic level. So I want to now shift gears and investigate this DNA a little bit more. So what is information? Information is the communication or the reception of knowledge or intelligence. It's also the knowledge that's obtained in like investigational study or instruction. It's intelligence, it's news, it's facts, it's data. The point of that is that information implies intelligence. There is some intelligent information to communicate. Okay, that's what information is by definition. So, I have no idea what is happening.
It's working.
Oh, wow.
I don't know why it just went to a link like this here.
Okay, who Googled Darth Vader?
Were you looking up?
I have no idea what's going on.
It's just scrolling through your iPad.
Well, I can see that, hon.
Do you really think the devil wants you to be teaching all this?
Yeah.
Don't you think if you were the devil that you would probably do everything you can to mess you up?
Okay. So, so point of that is information implies intelligence. I mean, it's what it is. So I want to just give a little illustration here. So Evan is off at college at Purdue, and he is now getting exposed to the humanist philosophies of life. That this first life came about through chemical evolutionary means. And I want to illustrate this because the other day, Terry decided to give a message to the kids through some cereal. So she took the cereal, and we got kind of a messy, cluttery area, and there's all these letters. And Evan walks down because we're all done and gone, and he's a teenager in college, and he's getting up 3 hours later than everybody else. And if you look up there, he sees this. He says, Christina must have been eating cereal because look at that big mess. And he walks by, he says, what on earth is that? And he's saying, and there's a little message in there. And he looks at it, and it says, take out the garbage. Now, Evan sees it, and he says, well, I'll be darned. Well, they just taught me in my classes at Purdue that I don't have to have an intelligent explanation for this. It is possible that if Christina spilled cereal enough times, that out of one of those times, a message like take out the garbage, Mom, can happen. So that must not apply to me. And by the way, it's really inconvenient for me to take out the garbage because I got a date with Sarah and I'm late. So he doesn't take the garbage out and leave. Next time, Elizabeth comes down. And she looks at it and says, that Christina is at it again. She never cleans up her messes. And she says, and that's a gluten cereal, so that certainly doesn't apply to me. And off she goes to meet up with her friends. And if you run across, and we used this example last week, if you run across, and of course it's intelligent science, I'm an intelligent being, and it's from Mom, clearly. So if you run across this, nobody would look at this and say that natural processes like the wind and the waves and the cutting of the sand could possibly produce this. We would look at that and say it's got specificity, it has got complexity, and it has got a message. Will you marry me, Nancy? And if you look at that, you would apply an observation, a scientific analysis of it, and say that this was not naturally made. Okay? You would have to be insane to look at a message like take out the garbage, Mom, and say that, well, it's possible that through natural means of spilling the alphabets that a message could just appear like that. What is silly about that is you don't get message. You don't get information from non-information. You don't get intelligence from non-intelligence. Okay? You don't get specified complexity from natural processes. That's not what we observe happening in the real world. So what happens is we now can look inside the cell. And what we see inside there is amazing. Watson and Crick get the name, recognition, and the accolades for discovering the double helix nature of DNA. But that is not where it all started. The fact that DNA had its involvement in heredity and in passing on the traits and characteristics was recognized a couple of decades before Watson and Crick. However, these suspicions about its involvement really came to fruition when we understood the structure. Why is that? Because the structure is perfect for passing on the information. We just looked at translating protein, but that is how it can replicate itself by these pairs that recognize each other. It can split apart and replicate itself perfectly. And so with the structure of the double helix, the understanding of modern era genetics is born. Now, what we discovered, though, is that the letters of DNA code for a very specific set of information. You see, once we discover the structure of DNA, and this is Edwin Chargraff in 1971, this discovery almost abruptly appeared to foreshadow a chemistry of heredity and moreover made possible the nucleic acid character of the gene. Avery gave us the first text of a new language, or rather he showed us where to look for it, and I resolved to search for this text. He's talking about a biochemist named Avery who really discovered the properties of DNA before they understood its shape. This is in the 1940s, okay, where he's talking about nucleic acids, how they match up, without a full understanding of exactly how DNA did what it did. That came with Watson and Crick. But he recognized that the nucleic acids are the text of a new language. It is an alphabet. is an alphabet that codes for a language, a chemical language in this cell. And that's why DNA is so important. So this is kind of the structure of DNA. It's this twisted ladder. It's got 4 letters to its alphabet. It's got an A, a T, a G, and a C. That stands for adenosine, thymine, guanine, and cytosine. By the way, DNA stands for deoxyribose nucleic acid. So that tells you there are 3 parts to a DNA strand. One is the deoxyribose. That's represented by this blue part here. That is a carbohydrate. Okay, it's a sugar. The P is a phosphate. Okay, deoxyribose phosphate. And then the A is a nucleic acid. Okay, thymine is a nucleic acid. Adenosine is a nucleic acid. Okay, so that's where we get deoxyribose nucleic acid, DNA. Okay, so there's 3 parts to each little segment of a DNA molecule, and that stretches on and on and on in the chromosome. So inside of DNA, you have an alphabet. T, A, G, and C. And that alphabet is put together in different ways to create a language that codes for every cell, protein, process, and structure that occurs in this incredibly complex set of machinery.
Well, in every living thing.
In every living thing,
Even
all the way down to the most basic life forms, the amoebas, the bacteria, even in viruses, okay? You've got these nucleic acids that code for its perpetuation and replication. So here's the DNA 4-letter genetic alphabet, and the importance here is that these things are always paired. Adenosine and thymine are always paired together. Cytosine and guanine are always paired together. So each strand of that double helix, each strand has got just a series, and then it's got its little partner next to it, and that's what makes it reproducible. It always matches up, because these things always match together. It's how they fit together perfectly to get into this double helix structure. So the point of this is that there is an alphabet that is the letters that code the information for this cell. The amoeba's DNA is a message. The most basic life form has got this message. And it is the equivalent of take out the garbage, Mom, okay? We would not look at take out the garbage, Mom, and ever think that that just came from being spilled naturally. It is a language. It is letters. They are put together in a particular, specific way to convey information. Take out the garbage. An intelligent being made that. The amoeba, there is an alphabet that has a message. And I ask you, why is it that we would say in all cases where a message is conveyed, there's an intelligent being behind it, but in the case of the cell, there is a very big, complex message that we would say just accidentally happened. So, first of all, DNA's 4-letter genetic alphabet. DNA of one human cell has the equivalent information of about 5 million pages of information, of 25,200-page books. So you walk into the library, they've got an average book of 200 pages. There's 25,000 volumes. There's 25,000 books in there. That is the information content of one cell. All of that's crammed into a nucleus that's of the cell, which is about 1/100th the width of a human hair.
Ooh. Whoops.
That was a really good one.
I know, what did that say?
And so, first life, the origin You see, all we ever see is life coming from life. We have never observed spontaneous generation, life coming from non-life. Experiments have been done, we have looked at it, and it just has never been observed. And furthermore, there is a high degree of specified complexity, message, language that implies, just through observation, that there is an intelligence that is behind first life. Because in the simplest cells, it is equal to about 1,000 volumes of an encyclopedia. That is from Richard Dawkins, who is probably one of the most prominent of today's voice of atheism. He is a biologist at Oxford. So, once again, going back to the beginning, what you have to believe, if you believe in spontaneous generation, that first life came about because of a large primordial soup where chemicals are formed from lightning and heat and thermal energy and electrical forces and heat vents and all kinds of stuff, that first life would come about with message, highly specific processes, power plants, and all this stuff would come about through undirected, unintelligent, that you could get message and information from unintelligent natural laws.
That takes a lot of faith. Yeah.
So you've got 1,000 volumes of encyclopedic information that just happened to assemble itself in just the right way,
For no reason.
to leap from non-life to life.
For no reason at all.
I want to remind you of the principle of uniformity. We looked at this last week. We can look at Mount Rushmore and we can see that an intelligence made that. We observed that happen. We can look at the Grand Canyon and we can observe the erosive properties, the geologic formation that happens through natural means. And we can compare them. We can see two types of causes. We can see intelligent causes like Mount Rushmore and natural causes like the Grand Canyon. And when we ask what caused an unseen past event, we use that principle of uniformity to look at the past event and say, well, the processes that make that stuff today is what makes them in the past. That is that principle of uniformity. The point is, what we are applying is a science of observation. What do we see happening? When we apply that, what we see is that when we have message, when we have specified complexity, we never observe today natural laws creating specified complexity. We don't observe that happening. We are simply applying science to make that conclusion. We are not applying our theology to make that conclusion. So, what do scientists say about all of this? Well, first of all, they say that life looks designed. Richard Dawkins, biology is the study of complicated things that give the appearance of being designed for a purpose. He wrote a book called The Blind Watchmaker. We talked about the watch analogy. And what he would say is, because of evolution, things have the appearance of design, but, well, that's all of it right there. It appears design, it just isn't. That's it. It appears design, but it isn't. Francis Crick, one of the co-discoverers of the double helix of the DNA process, biologists must constantly keep in mind that what they see is not designed, but evolved. What's interesting about this...
Very hard trick to do.
What's interesting about this is he says we have to constantly remind ourselves, because He doesn't want you slipping into thinking about the fact that this is a really highly designed system that implies a designer. He says, keep remembering that this is evolved, not designed. You have to remember that it's not designed, but it's evolved, because it's really hard to do because it looks so designed. That's what he's saying. And he also said the origin of life appears to be almost a miracle. So many are the conditions which would have had to be satisfied to get it going.
Almost.
The point is, here is the acknowledgement that I have to suspend my scientific belief to latch on to the fact that you could get this highly specified, complex, designed information system from completely natural laws. I have to keep reminding myself, this is evolved, this is evolved, this is evolved, because it looks so stinking well designed.
It seems so contradictory.
Look, and here's the final thing. Everything depends on itself. Okay? DNA codes for proteins, but it uses those same proteins to make itself. DNA depends on proteins, proteins depends on DNA. DNA and RNA. DNA is the message behind the RNA, but it takes the RNA to make the DNA. Well, the RNA is what has the message for all the processes of the cell, but the apparatus of the cell is completely necessary for the RNA to be made and perpetuate itself. Well, and the proteins are the substance of the cell, but the cell is necessary for those proteins itself in the manufacturing. And the DNA needs the cell, and the cell needs the DNA, and the RNA has the codes for the proteins, and the proteins are necessary for. The point is, it all works in this ballet, this dance, all together. Any part without any other part doesn't exist. They all have to work together to perpetuate themselves. In first life, you need this entire complex dance working together to make it happen. You can't have just a couple of complicated proteins that say, hey, we'll make a little DNA today, because it's got no way of knowing how or what to do to put them together. Okay? The DNA is dependent on the cell and the RNA. The proteins are dependent on all of these. Everything is interdependent in this giant, highly designed and complex dance. So there are just simply insurmountable odds. Yeah.
Because evolution theorizes little tiny changes over time. But the chicken and the egg, the point of that would be what evolved first. How could you evolve DNA if you didn't have RNA already made to process it?
Right. That's exactly right.
How would you evolve RNA if you didn't already have proteins that could make the RNA? So all of that stuff had to be there at the same time for it to ever function at all.
Correct.
Otherwise, there's no evolutionary advantage to random DNA that doesn't have anything to work with.
That's right.
And there's no way that you could just evolve that gradually. It all had to appear at the same time.
Exactly. That's exactly the point, is that it all works together in that kind of symphonic dance. None of the pieces work without all the others.
Right, and even with that very first cell.
Exactly.
Whatever that first living cell was, all of that had to already be there for it to work at all. And then to be able to produce every other cell in the universe.
So everything is interdependent, and you can't get that by successive piecemeal. First you get some proteins, then you get a little DNA, then you get the RNA, because it all depends on each other to keep it going. So there's no way to go from an egg to a chicken. It's got to have it all together. The chicken and the egg come together. So anyway, the insurmountable odds, no matter how large the environment one considers, life cannot have had a random beginning. There are about 2,000 enzymes, and the chance of obtaining them all in a random trial is only 1 part in 10 to the 40,000. An outrageously small probability that could not be faced even if the whole universe consisted of organic soup. If one is not prejudiced either by social beliefs or by a scientific training into the conviction that life originated on the earth, the simple calculation wipes the idea entirely out of court. And the enormous information content of even the simplest living system cannot, in our view, be generated by what are called natural processes. That is Hoyle and Rick Mashinghi. So a common sense interpretation of all of this is of the fact suggests that a super intellect has monkeyed with physics as well as chemistry and biology, and that there are no blind forces we're speaking about in nature. And that's astronomer Fred Hoyle who said that. And I wanted to speak a little bit about the question that came up that I said I'm going to get to that at the end. Okay, what is this all about? All right, and this is going to take me about 5 minutes, and we're at the end of our time, but I think it'll be worth just kind of going through this as a conclusion. I want to talk a little bit about the whole creation-evolution debate, and I want to tell you guys, it is not about looking at Darwin and saying, that is so dumb, how come people stick with that? It's not, Darwin is brilliant. Darwin is a super intellect, okay? He made observations that have greatly advanced science and our understanding of geology and biology. And so it is not something to just poo-poo the theory. But I'm going to get to what is behind the latching on of Darwinian evolution in this little discussion here. But when you go on the web and you look at the vitriol against the type of stuff that we're talking about today, it is painted as this creation evolution or intelligent design versus naturalism. It is painted as a war between religion and science, or it's painted as a war between faith and reason. And the common epithet is, here comes those crazy religious fanatics, fundamentalists, to impose their dogmatic religion on everyone and ignore science. These guys are using bad science, ignoring the real stuff, and man, just look at this garbage that they're trying to give us. Don't you listen to intelligent design, because that's just repackaged creationism, which belongs in the 1600s, and why do these people keep doing this to us? Mm-hmm. I had a quote on here, I took it out from a blog post that was just pure vitriol with nothing intelligent in it. And that's how it's presented. But actually, this creation evolution debate is a debate about good science versus bad science and reasonable faith versus unreasonable faith. That is the true crux of what this debate is about. So I wanted to take a look at that, and I wanted to point out that there is a fundamental philosophic bias against the supernatural and against the idea of something outside of the physical realm causing the cosmos or life. Okay? And this quote in my slide series was inadequate. So I went back to this guy here, and I'm going to tell you that stuff by Dawkins and this guy, this type of language is used. Okay, this type of language is used. I did not just get the most incendiary quotes. I'm telling you, this stuff is out there. Okay, so this is the bias that we're dealing with, and I want to go through this slow because it's there's a lot of big words. Okay, but I want you to just wrap your minds around what this guy is saying. Our willingness to accept scientific claims that are against common sense. We accept claims that are against common sense. Is the key to the real struggle between science and the supernatural. Intelligent design versus naturalism. Okay? Our willingness to accept scientific claims that are against common sense is the key to the real struggle between science and the supernatural. We take the side of science in spite of the patent absurdity of some of its constructs. In spite of the tolerance of the scientific community for unsubstantiated just-so stories. What's a just-so story? Well, we had non-life, it came to life, that's just so. We've never seen it happen, but that's just the way it is. Because we have a prior commitment to materialism. In other words, our philosophic bias is to materialism. Therefore, because we have that, we will go against common sense. We will go in spite of patent absurdity of some of this stuff. Because we have a commitment to materialism, we're going to stick with it, even if it's absurd. Against common sense is the key to the real struggle between science and the supernatural. We take the side of science in spite of the patent absurdity of some of its constructs. in spite of the tolerance of the scientific community for unsubstantiated just so stories, because we have a prior commitment to materialism.
So basically, they decided in advance that God can't exist. Therefore, there's no intelligent designer, no matter how designed the universe looks.
And he's going to make that,
Period.
he's going to say that
That's
himself.
Right.
He's
There
going
you
to say it himself. It is not that the methods and institutions of science somehow compel us to accept the material explanation of the phenomenal world. So it's not what we observe that compels us to do that. It's not what we see in science. There's nothing about science that compels us to take a materialistic, naturalistic view of things. It's not the methods of science that somehow compel us to accept the material explanation for the phenomenal world, but on the contrary, that we are forced by our a priori adherence to material causes. We're forced by our a priori adherence to material causes to create an apparatus of investigation and set of concepts that produce material explanations, no matter how counterintuitive, no matter how mystifying. So even if what we're doing is counterintuitive and mystifying because we have an a priori adherence to material causes, moreover, that materialism is absolute, for we cannot allow a divine foot in the door. We have a priori rejected an intelligent design being. God cannot be allowed in the door. Therefore, even if what we're doing is just so causes, even if it looks completely absurd, even if it's not common sense, we stick to it because we are sticking with materialism. Now, I ask you, who's dogmatic? How many verses of the Bible have I quoted to you to prove my point? How many? None. How much science have I given to you to prove my point that there is specified complexity in design in the universe and in first life? Tons. Who's the dogmatic one? Hubert Yoke is a physicist and information scientist. Okay, he said the belief that life on Earth arose spontaneously from non-living matter is strictly a matter of faith, in strict reductionism, and is based entirely on ideology. What he's saying is people that stick to that are the ideologues. Okay, reductionism is the idea that life can be boiled down to its non-living chemicals. That's what reductionism means. And so observation and reason shows that life has complex specificity. It's got design pieces to it. That it's got message and text. And that it's got this irreducible complexity. Irreducible complexity is something I'm going to talk about next week. Okay? And it's that there are certain processes that have a minimum number of pieces that you cannot evolve. Because any part of this thing without the other parts cannot exist. In other words, there are things that you reduce it down to its 10 basic things, but without all 10, it doesn't work. So you can't make it piece by piece by piece by piece, because without all 10 pieces, it doesn't work. That is irreducibly complex. So, that's it. That's my case.
That's all you got?
All right.
The faith that it takes to believe in spontaneous generation requires the kind of faith that it takes to believe that you can get a 1,000 volume encyclopedic information by blowing up the print factory enough times. If you just blow it up enough time so that the ink and the paper mesh in just the right way, that out of that you could conceivably and possibly get this brilliant 1,000-volume encyclopedia.
Analogy I heard that I love too is it'd be like if a tornado came through a junkyard and assembled a 747.
If you get enough tornadoes and enough junkyards, who knows? All right, thanks guys. We'll see you next week.